So I'm going to present the results of a study conducted at INRS with this title, Metabolic and some endocrine disruptions following gestational exposure in rats to organophosphorus flame retardants using blood biochemistry and fatty acids concentration and profiles. So first, a brief introduction. Flame retardants are molecules that prevent or slow down the start of fires. And they can be found in a wide range of industries, including electronics, construction, automotive, furniture, and textiles. Following the discovery of the toxicity of certain substances, like brominated compounds, European regulations have evolved to limit or ban their use. So organophosphorus compounds are the OPFR, are the substituents that have come onto the market. They are found in the indoor environment in the form of dust or airborne particles, and they are emitted during production, wear, or disposal of finished products by abrasion, volatilization, or leaching processes. Human exposure has been demonstrated by the presence of OPFR metabolites in urine, breast milk, placenta, seminal fluid, etc. In the literature, several epidemiological studies and experiments using rodents or in vitro models have shown that OPFR have effects on reproductive function, on fertility, on steroid receptors and synthesis pathways, as well as on metabolism. Triclosyl phosphate, TCP, which is one of the OPFR analyzed in this study, is persistent in the aquatic environment, inhibits fertility, and accumulates in the livers of Japanese medaca larvae. In our study, to demonstrate the effects of gestational exposure to TCP, we exposed pregnant rats from day 12 to day 19 of gestation by oral gavage to 4 different doses of TCP. At gestational day 19, organs and biological fluids were collected for analysis. First of all, molecular analysis using candidate gene approach with RT-qPCR was performed, and the expression of genes involved in the steroidogenesis, cholesterol metabolism, and fatty acids metabolisms was analyzed in maternal liver and adrenals liver, and adrenals mother, and also in the liver, adrenals, and testes of male fetuses. But today I will present only the data for the pregnant females. This is followed by a metabolic analysis using biochemical analysis of the mother's plasmas and fatty acid profiles in the liver, adrenals of the mother, and also on the livers of the fetuses, male and females. And finally, for the endocrine disruption, we performed a hormonal analysis on the testes of the male fetuses to identify any disturbance in the fetal testosterone production. So first, for the results, I will present the maternal physiology data. So TCP induced a slight maternal toxicity at the highest dose, in red, with a decrease in absolute weight gain, a decrease in the liver to body weight ratio, here, and an increase in adrenals to body weight ratio. After, for the gene expression data in the maternal organs. So I resume the RT-qPCR results in the heat map. So you can see here the biological pathways, the genes involved in these pathways. First, here in the left, the result for adrenals, and here on the right for the livers. D1 to D4 is for the doses to the lowest from the highest. And a red box is overexpression, and a green box is a genes we have a decrease as a decreased expression. So here we can observe that exposure to TCP induced disturbance as early as the low dose for certain genes. And we also observe different gene expression profiles between the two organs. So here, TCP induced a disruption in the expression of genes which involved in cholesterol and lipid metabolism in the mother's organs. So we continue with the metabolic analysis. TCP induced a disturbance in blood biochemistry at the highest doses with an increase in the liver enzymes concentration, a decrease in the glucose, and in cholesterol concentration. TCP here, therefore, has a metabolic disruptive effect on the mother. I'm going to present the results for the fatty acids assays. TCP did not affect the concentration of fatty acids in maternal blood. However, we did observe an increase in neutral lipids, which represent the lipid storage in cells, and phospholipid concentration, which represent the membrane, in the liver of the pregnant rats, here. Also, the concentration of neutral lipids was also increased in the mother's adrenals after exposure to the highest dose of TCP. So these results implied an increase in lipid storage in maternal liver and adrenals, as well as a structural disruption of the liver only. In fetuses, there was no change in phospholipid concentrations in male and female liver, but there was a significant increase in neutral lipid concentration following exposure to the highest dose of TCP. And this increase was more marked for female than for males. TCP therefore induced a metabolic disturbance in the fetuses too. Here, for information for the endocrine disruption, TCP induced a slight decrease in fetal testosterone production at the highest dose of TCP. To conclude and give a biological meaning to all this analysis, first of all, if we look at the maternal adrenals, we demonstrated that exposure to TCP induced an increase in maternal adrenal weight at the highest doses, associated with a disturbance in blood biochemistry and an increase in fatty acid storage. TCP induced increased expression of genes involved in cell entry and de novo synthesis of cholesterol. And in particular, the increased expression of PEROSH gamma gene, which has a major role in fatty acid absorption and storage in cells. All these results are in line with the literature, where it has been shown that in non-pregnant rats, TCP exposure induces lipidosis due to an accumulation of cholesterol esters in the adrenals and disturbance of blood biochemistry without alteration of reproductive cycles. Here we have demonstrated similar results, but in pregnant in pregnant female rats, additionally observing disruption in the genes involved in these pathways. With regards to the liver of the pregnant rat females, TCP induced a marked disturbance with a decrease in liver weight at the highest dose, associated with an increase in blood concentrations of hepatic enzymes, indicative of hepatotoxicity. In addition, these disturbances were associated with an increase in concentration of neutral lipids and phospholipids, indicative of an intracellular lipid storage and a structural disruption. In addition, pas du tout. These physiological disturbances were associated with molecular changes, in particular with increased expression of SCD1 gene, which encodes a key enzyme in de novo lipogenesis in the liver. Its overexpression has been shown to induce lipid storage in the liver, as it plays a central role in the biosynthesis of monounsaturated fatty acids from saturated fatty acids. These effects resemble the symptomatology of non-alcoholic fatty liver disease, where patients present a weight loss, an increase in serum hepatic enzymes concentration, hepatic lipids accumulation, and structural disturbance of the liver. So, TCP induced also metabolic disturbance in the fetal liver with an increased lipid storage at the highest dose of TCP. However, we cannot dissociate the direct effect of TCP on the fetus from an indirect effect via the maternal metabolic disturbance induced at the same dose. However, this metabolic disturbance was observed in the fetuses, and it could have long-term consequences after birth. So in conclusion, we have demonstrated a slight endocrine disruption associated with maternal toxicity, and TCP induced also metabolic disturbances in the mother and in the fetuses. We performed the same type of experiment on two others of PE4. There was a mistake here, it's not triclosyl but triphenyl phosphates, TPHP. And the second one, I'll let you read the entire name, TDCPP. And we can conclude that the three analyzed OPA4 present different toxicological profiles. Their industrial benefits must therefore be carefully considered in the light of their toxicological properties. And it should also be noted that occupational exposure tends to be to mixtures, and that the toxicological profiles of mixtures may differ from those of the single molecules. So it could be relevant to determine through a field study whether metabolic changes are also observable in employees exposed to these chemical agents and the consequence on the health. So thank you for your attention, and if you have any questions.